A-Level Biology 9700, Paper 4, Variant 1, October, November 2024. And question number 5 is Lunularia cruciata is a primitive plant. Its body consists of a flattened sheet of photosynthetic tissue called thallus. Thallus, you can say the vegetative part of the plant, which is not differentiated into roots, stem, or leaves. So, thallus is the photosynthetic tissue. So, the photosynthesis takes place by this part of the plant. Now, figure 5.1 shows L. cruciata with two different types of reproductive structures labeled A and B on its surface. Structures A and B and the thallus of L. cruciata are haploid. And this thallus is all found in some type of algae, fungi, and in liverworts. So, Lunularia cruciata is a liverwort. So, this is a liverwort. So, they lack roots, stems, and leaves. So, in this way, I would like to say, first of all, the question is explain what is meant by haploid. Haploid means cell or organism. You know, all organisms, whether they are animals or plants, they have nucleus. We are talking about plants. So the plants, they have cell wall, cell membrane. Inside these, they have nucleus, and inside the nucleus, chromosomes chromosomes are there. A set of chromosomes is there. And here you know that students, okay, these chromosome number is specific for each species of plants and animals as well. So here we are talking about the liverworts. The liverworts range from 8 to 9 chromosome number in different species of these liverworts. So here in this case, or Lunaria cruciata, the chromosome number can be the 8. So 8 means they have 8 number of chromosomes, or if we make their pairs, so 4 and 4, there will be a complete set of chromosomes. But when we say they are not pairing with one another, a single set of chromosomes, it means 4 number of chromosomes, unpaired chromosomes, which are present, and N. When we say 8 number of chromosomes, we say 2n number. 2n number is called as diploid number. And when we say they are in the form of single set of chromosomes, they are represented by single n, it means haploid. Haploid means half number of chromosomes or a single set of unpaired chromosomes. So we can say in sperms or in eggs, which are the gametes. Gamete cells, which are sperm, which is present in male, eggs are produced by the female. So haploid number of chromosomes are there. It means half number of chromosomes or single set of chromosomes which are present in sperms and eggs. Because during the sexual reproduction, sperms fuse with the egg. And if the sperms have 4 chromosomes, and egg will also have the 4 number of chromosomes, when there will be the formation of zygote, the chromosome number will be maintained, which is the characteristic of that species. That is, if already these sperm and eggs have 8 number of chromosomes, and in that case, you can say the chromosome number of that species will be changed, which is not possible. So in this way, haploid is the half number of chromosomes or a single set of unpaired chromosomes. So it was all about this. We have to explain only, though they have asked that what is meant by haploid. So we have to explain only what is haploid. Next question number B is there. Structure A contains pale discs of tissue C that can germinate into new L. cruciata. The new plants that develop from C are genetically identical to the parent plant in figure 5.1. So structure B, first of all, we will discuss what is about this tissue C that can germinate into new cruciata. The new plants that develop from C are genetically identical. Students, two types of cell divisions are there, that is mitosis and meiosis. During mitosis, the daughter cells, when the cells divide, the daughter cells which are produced, they are genetically identical to the parent cells. Because in this way, the mitosis is that type of cell division in which the chromosome number is maintained generation after generation. That is the every cell, because mitosis takes place in the somatic cells, so the chromosome number should be maintained. So during mitosis, the offsprings, or you can say the daughter cells which are produced, they have the they are exact copies of their parent cells. So in this way, we can say the genetically identical. So it means C mitosis. Here the mitosis will take place. And now we will move towards the structure B contains male sperms that are chemically attracted to swim to female eggs on a neighboring parent plant. B, male sperms means the gametes, sperms, male gametes, eggs, female gametes, male gametes and female gametes. So, in sexual reproduction, already told you in the previous question, the male gametes unite or fuse with the female gamete. So, male gametes have haploid number of chromosomes and female have or haploid number of chromosomes. So, when they fuse together and zygote is formed, then there will be the formation of complete set of chromosomes. So in this way, again, the chromosome number is maintained generation after generation. So meiosis takes place only in gamete cells, not in the somatic cells. So mitosis takes place in the somatic cells and meiosis takes place in the gamete cells in which this is the reason it is also called as the reductional division, because the daughter cells, which are produced as a result of meiosis, all they will have the haploid number of chromosomes instead of diploid number of chromosomes, because as a result of this meiosis, gametes are formed, sperm cells and egg cells are formed. So I think it's enough. So structure D develops to produce. Structure D will be the zygote to produce spores that grow into new plants that are genetically different from the genetically different means again we have a proof that the meiosis has been taken place, and genetically why they are genetically different, and in the meiosis, in the prophase burst of meiosis, the chromosomes exchange their parts. During crossing over, they join to the homologous chromosomes come close to each other and they exchange their parts. Due to exchange their parts, and then as a result of this exchange of their parts, the variations are originated. Variations are originated, and so they will be genetically different from one another. So it was all about the question. Now, what they have asked, identify which of the structures A, B, C, and D associated with sexual reproduction. C was associated with mitosis, in which asexual reproduction. Asexual reproduction, because all daughter cells are produced as a result of mitosis, they will be genetically identical. So in C, and then the sexual reproduction in B, because these are the gametes. So B and D both are associated with sexual reproduction and then produced by mitosis or asexual reproduction, that is C. The site of meiosis, already told you, in the B, they are gametes, the meiosis takes place over there. So each letter may be used once, more than once, or not at all. So it was all of this question, B. Now we are moving towards the next question, and next question we can see a figure 5.2. Equus cabellus horses are haploid animals that produce sexually. Male and female horses produce gametes which fuse to form genetically different offsprings. Already told you that as a result of meiosis, the chromosomes exchange their parts, and as a result of exchange of those parts, the chromosome um, get separate from when they get separate from one another randomly. So they they will be genetically different from one another. So in this way, I would like to say, question, explain the need for a reduction division during meiosis in the production of gametes in animals such as horses. In all type of organisms, this is very compulsory that there should be the reduction division in gamete cells, as you know, because if uh, every species has chromosome number, a specific chromosome number. What happens if the chromosome number is changed? The species characteristics will be changed. So in this way, I would like to say these forces, uh, they have diploid number, two sets of chromosome are there when they have diploid. So all somatic cells will have the diploid number of chromosomes. Diploid means two sets. And then sexual reproduction involves the fusion of two gametes, sperm plus eggs. So what will happen with them when they will fuse together? If they have 2n diploid number, 2n diploid number. So the zygote which will be formed as a result of sexual reproduction, the chromosome number will be 4n, which is not the specific species characteristic. So it cannot happen. So only what happens, the reductional division, if there is reductional division, as a result of this reductional division, the when these sperm and eggs fuse together with a haploid number of or single set of chromosomes, then they will produce a 2n number of chromosomes. It means zygote is further changed into embryo, and then embryo into fetus. So in this way, the different developmental stages takes place to become a fetus and then a baby horse. So in this way, the zygote must have 2n number of chromosomes. But if the gametes already have diploid number of chromosomes, so there will be the problems because this chromosome number of the species will not be maintained. It will be changed. So that species will not exist in any way. So maintains the species character. So the chromosome number or this reductional division or the maintains the species characteristic chromosome number generation after generation or cross generation. So generates genetic diversity through the shuffling of genetic material, contributing to the adaptability of the species. So in this way, um, um, the chromosome number should be reduced in the gamete cells, because it will it will help to maintain the chromosome number of that species. So in this way, the meiosis play a crucial role, because during the uh, meiosis, the chromosome is number is reduced to half, because it takes place in the gametes. Now we are moving towards the question number 6. The mammalian kidneys are responsible for the excretion of urea. They are responsible for the excretion of urea and osmoregulation. So homeostatic control of water potential of the body. So first of all, what is excretion of urea? Excretion of urea is actually the removing of urea. What is urea? A nitrogenous waste product. Urea is a nitrogenous waste product which is harmful if remain inside the body. So it should be excreted. And secondly, the osmoregulation. Osmoregulation. So these are two major functions of the mammalian kidney. Osmoregulation, the homeostatic control. So what is osmoregulation? Is actually the maintaining the balance, maintaining the balance of water and salt in the body fluids, in the blood or body fluids. So in this way, uh, question is outline how and where the excretory product urea is made in the body. Urea is where it is excretory product urea, nitrogenous waste product which should be excreted out of the body. It is the main function of the kidney, that is osmoregulation. So during the process, it should be excreted. How and where? So urea is made in the liver through the deamination of amino acids, which produces toxic ammonia. The liver detoxifies ammonia by converting it into urea by the urea cycle. Urea is then transported to the kidneys for excretion of urine. So the answer which I have written, now I would like to explain how the urea formation takes place. Actually, the urea is primarily produced in the liver of the mammals. So when proteins are broken down, proteins, when the proteins are broken down into, when proteins are broken down, they will changed into amino acids. After breakdown, so they will change into amino acids, and in the liver these amino acids undergo process called deamination. So deamination takes place in liver, and these amino acids will produce the deamination means removal of amino group, removal of amino group from the amino acid. And as a result of deamination, there is formation of ammonia. Ammonia, and the formula for ammonia is NH3. So ammonia is highly toxic to the body. So what happens to detoxify ammonia? The liver converts it into urea. Liver will convert ammonia into urea through a series of enzymatic catalyzed reactions known as the urea cycle. So this urea cycle combines ammonia with carbon dioxide to form urea. So ammonia uh, will well done. So there will be the formation of urea. So we know that urea is then transported from the liver to the kidneys via the blood from liver to kidneys. and in kidneys the process takes place and it is excreted out of the body in the form of urine. So I would like to say that the kidneys filter urea from the blood and excrete it in the urine. So urea is made, I have written this, that urea is made through a delamination of amino acids, which produces toxic ammonia, and then ammonia is detoxified by the liver and converted into urea. So urea is then transported into the kidneys, and then from kidney, it is excreted out in the form of urine. Next question. Identify the names and locations of these components of homeostatic control in osmoregulation. So what are those? Uh, Let me move back to the question. Homeostatic control of the water potential of blood includes receptors, effectors, and target cells. So identify the names and locations of these components of homeostatic control in osmoregulation. First of all, I will go with the receptors. So receptors, we can also say them the osmoreceptors. So they are also called osmoreceptors. So they are located in the hypothalamus. What is hypothalamus? Hypothalamus is the part of the forebrain. Part of forebrain. So it is, you can see forebrain, part of the forebrain, which is also called as thrust center, because it controls the osmotic pressure, thrust center. And second, after these receptors, there are effectors. So effectors, we can say that they can be the kidneys because they are affected by this, kidneys or effectors. Effectors are the kidneys. So the kidneys, and especially we can say the part of the kidneys are the collecting ducts. So they are located in the, you can say, kidneys are located in the lower back, one on each side of the spine, and then third part. And what is third part? And third is the target cells. So the target cells, any part of any cell of the body can be the target cell. So can be present throughout the body, can be present throughout the body. Now, how it works. Now, after this explanation, I would like to move how these three different types of takes place, how it works. So first of all, I would like to say the osmoreceptors, which are the receptor cells. Osmoreceptors. Osmoreceptors. How do it take place? How it works? Osmoreceptors in the hypothalamus, in hypothalamus detects changes in the water potential. Changes in water potential. What is the function of receptor cells? They detect the changes. Receptor cells will always detect the changes. And in this case, the osmoreceptor cells, they will detect the changes in the water potential. So they will detect the changes in the water potential. And then what will happen? Of water potential of blood, and then we can write in the form of points. Secondly, if the water potential is low, there will be the difference. If water potential can be low or water potential can be high. If the water potential is low water potential, we can say low water potential, what can happen at that time? So at that time, when water potential is low, the hypothalamus triggers the pituitary gland to release antidiuretic hormones. ADH will be released. It will be released by pituitary glands. So what will its effect? So they will act on the collecting ducts of the nephrons in the kidney, making them more permeable to water. This allows more water to be reabsorbed. More water will be reabsorbed. If water potential is low, ADH will be released. ADH means a hormone which is secreted by the pituitary gland. It is responsible, or it controls the water potential inside the body cells. So what will happen at that time? Antidiuretic hormone will be secreted, due to which more water will not excreted out. Most of the water will be reabsorbed because there is less water potential inside. The water potential and it should be maintained. It should be regulated inside the kidneys. So to protect the body from dehydration, less water is excreted out. Most of the water is reabsorbed. On the other hand, if the water potential is high water potential, so what will happen? The most of the less ADH will be excreted out, due to which the most of the water will be excreted in the form of dilute urine. Less water will be absorbed because the body cells do not need more water at that time. Already water potential is very high inside the blood, so most of the water will be excreted out in the form of dilute urine. So in this way we can say the inside the body, the osmoregulation or the control will take place inside the body. So this results in less water reabsorption, and dilute urine will be produced. So in this way, the whole process will take place. These factors and first of all, the receptor cells, then effector cells, and the target cells, they will work together for the homeostatic control of the water potential of the blood. In both cases, if water potential is high or water potential is low, so in both cases, uh, the process will take place. If water potential is low, more water will be reabsorbed, and less water will be excreted out in the form of thick urine. But in other case, if there is high water potential, most water should be excreted out. So at that time, less KDH will be produced by the pituitary glands, and most of the water will be excreted out in the form of thin urine or dilute urine, you can say. Now we are moving towards the next question. And next question is, The glomerulus and Bowman's capsule of the nephron are important in the formation of urine. Outline the role of the glomerulus and Bowman capsule in the formation of urine. First of all, uh, the main two processes which take place in the kidney, in the nephron, they are fine filtration or ultrafiltration. Fine or ultrafiltration. And second is selective reabsorption. So these are the two major parts which are performed by the nephrons. So here you can see the glomerulus and Bowman capsule. These are the part of the nephrons. Glomerulus, a tuft of cilia, which works as a filter. The blood is filtered through this glomerulus, and after filtration, the filtrate enters into a cup-shaped structure, which is Bowman's capsule, which is after the name of a scientist, Bowman. So this is the Bowman capsule or cup-shaped structure in the nephron. So glomerulus help in the filtration, ultrafiltration, and the filtrate is collected into the Bowman capsule. Why it is collected in the Bowman capsule? Then it passed through a process. And then you can say when the filtration takes place, all there are different, you can say it filters out small molecules based on size and pressure. So during this high pressure filtration, uh, water, small molecules like glucose, amino acids, and waste products like urea out of the blood they come into the filtrate. As for example, I would like to draw a little uh, to explain you. So this is the cup-shaped structure, and inside the cup structure there is glomerulus, and from glomerulus, when after filtration, so blood enters into the ultrafiltration takes place over here, and as a result of ultrafiltration, uh, the filtrate. So here will be the filtrate, and in the filtrate there will be the water, glucose, amino acids, ions, and waste products in the form of urea, etc. So all they will be in this filtrate. So it means in the previous question, I have told you if the water potential is low, the most of the water will be reabsorbed. So the reabsorption takes place in the part of the nephron after filtration, because you can see in the filtrate there are so many important nutrients inside. Glucose is there, amino acids there, ions are there, which should not be excreted out the body. So the process of selective reabsorption takes place. So as a result of the selective reabsorption, all these different nutrients and ions and water as well, which are important for the body, they are reabsorbed over here. So I will explain all this in this way. First of all, I would like to explain the Bowman capsule, the ultrafiltration, because after filtration, the filtrate enters into the cup-like structure. It surrounds the glomerulus. Its primary role is to collect the filtrate. Again, fine filtration by the glomerulus, and then then filtrate enters into the cup-shaped Bowman capsule. So Bowman capsule also acts as a funnel You can see I have drawn this a small drawing, directing the filtrate into the renal tubule for further processing. What is renal tubule? The different other parts of the nephron. So I I would like to say uh, and the sieve, we can also say a sieve to the glomerulus because it filters out small particles, water, small molecules from the blood and the cup-shaped umel. It will collect it. So in this way, the process takes place. Now I would like to read the answer which I have written. I will explain it as well. Glomerulus is a network of capillaries within the Bowman's capsule. It's yes, tuft of cilia or the capillaries, uh, which helps in the ultrafiltration. A fine filtration, a blood enters with high pressure into the glomerulus, and from the after filtration from the glomerulus, it enters in the form of filtrate into the Bowman capsule. So its primary function is ultrafiltration or fine filtration because it filters out small molecules based on size and pressure. So all which will be smaller in size, they will be uh, into the filtrate. So filtrate will be collected by the Bowman capsule, which is cup-shaped structure. So it surrounds the glomerulus. Its primary role is to collect the filtrate. So the fluid which is filtered out of the glomerulus is collected by the Bowman capsule, which is not urine, because it still contains valuable substance that need to be reabsorbed back into the bloodstream. So all they are already written over here, that is they can be the water, they can be the amino acids, they are glucose, ions, and waste products as well. So the selective reabsorption will take place. If the concentration is low, most of the water will be absorbed and less water will be excreted out. Amino acids will be reabsorbed, glucose will be reabsorbed. The ions which are needed by the body, they will also reabsorbed into the blood through the collecting duct. So in this way, the process uh, takes place into the uh, two parts of the nephron, the major two parts, because the major function is taking place over here. That is the ultrafiltration and selective reabsorption. All these two parts, these two functions are performed into the nephron. So these are the major functions. They have asked about this. Now we are moving towards the next part, which is C. And here you can see concentrated urine contains a high concentration of solutes and small volume of water. Different species of mammals vary in their ability to produce urine with a high solute concentration. So table 6.1 compares the ratio of the solute concentration of urine U to the solute concentration of the blood plasma P in some mammals species. The habitats of the mammal species are also shown. So table 6.1 is there. With reference to table 6.1 suggests the what the different values of U ratio P. U solute concentration of the urine and P is the solute concentration of the blood plasma. So about the ability of these mammal species to tolerate a shortage of water in their environment. So the different values of UP shows that I have written the answer first before going in this written answer. We have to show about the ability of these mammals. We have to discuss all these mammals one by one. First of all, UP value indicates how much more concentrated the urine is compared to the blood plasma. So we have to compare both of them. So a higher UP value means the urine is more concentrated. If there is solute concentration of the urine or solute concentration of the blood plasma, if this ratio is high, it means the urine is more concentrated and then reflecting a greater water conservation. Water should be conserved in that case. If there is, you can say, if there is urine is very thick. And then first of all we will move towards the uh, kangaroo rat, which has highest ratio, that is 16 is to 1. 16, it means 16 is for solute concentration and 1 is the solute concentration of the blood plasma. So urine is more concentrated as comparison to blood plasma. So it is the highest UP value. This is consistent with its desert habitat where water conservation is crucial. So that is the reason this value is high, because a habitat is desert, and less availability of water is there into the habitat. So the solute concentration of urine is high because of the less availability of water. So it is 16 as comparison to the solute concentration of the blood plasma. So it is the highest value in this case. And second highest value is in rat. And rat's habitat is variable. It rivers in all types of habitats. So it has a relatively high UP value, but less than the kangaroo rat. So you can see has a relatively high, which is interesting, lives in a variable environment. This suggests that it has a good water conservation abilities, possibly as an adaptations to potential water scarcity. So it can have this ability because it is living into the variable environment. And then we come to the next, camel. And here you can see the camel has, again, it lives in the desert and it has age is to 8 to 1 ratio. So in this way, high UP ratio reflecting its ability to conserve water in the desert, because it all has to conserve, again, non-availability of water into the desert. So it has to conserve more water inside. Then we come to the human beings. So we have, or the human beings have a moderate UP because we live in different types of habitats. So have a moderate UP ratio. So it reflecting our ability to adjust urine concentration based on water availability. Already discussed in this in today's video that if water potential is high, more or most water will be excreted out in the form of urine. So in this way, the human beings have the variable environment or variable habitat. So they have moderate ratio, UP ratio. And then after human, the least beaver. They live in rivers and lakes. It means they have the lowest UP value because they produces the least concentrated urine, because they are, you can say, they are living in the in a place, around, they have water bodies around themselves. They are living in the rivers and lakes, so they don't need to conserve water as much. So they are in 11 bodies, so they don't need, so they have the least one, which is 1.7 is to 1 ratio. So the different values of UP shows that species with higher UP ratios are better adapted to tolerate water shortages. As I've written this answer, so they have better adapted to the environment because they are able to conserve more water. They can tolerate shortage of water as well. So minimizing water losses. Species with lower UP values are less adapted to water shortages. They produce less concentrated urine. Definitely their urine will be thin if they have more water inside. If they have less water inside, there will be the thick urine. Less water will be excreted, or most of the water will be absorbed. The body needs to conserve the water, so most of the water will be uh, stored, or it will be reabsorbed into the body, and less urine will be excreted out, and thick, which will be a thick urine. more concentrated urine. And but in the with lower UP values, the species will don't need to conserve more water because they have or they are living in that environment where there is no need to conserve the water or to save the water. So their urine will be dilute and less concentrated. So the UP ratios effectively reflect the water conservation strategies of different mammals species directly correlation with their habitats and the availability of water. So their UP values depends on the habitats where they are living. They have to adapt according to that environment to live alive. So in this way, it was all about question number 5 and 6. Please like, share, and subscribe my channel. I have already given my email ID in the description. You can contact me in case of any problem. Thanks for watching.